What's up guys, good evening my chilla party. Well, this is your NSEJS biology master teacher and I welcome you all to the session number 2 for NSEJS 2025 biology basic to advanced series. Now guys, in the previous class we had started this unit, that is biomolecules. It is one of the most important units, or I should say the concepts that are there in your NSEJS syllabus. So first of all, I'll give you a brief summary what we had covered in the previous session, and those who haven't watched it yet, please go check it out. It's going to help you a lot building your basics about biology. Well, biomolecules, so I had told you that a cell is made up of over 5,000 chemicals. Then we had defined what is biochemistry, so the science dealing with these chemicals and the biochemical reactions occurring in a living organism and their life processes. Okay, so the study revolving around this is biochemistry. Then what are biomolecules? So these are the carbon compounds, the organic molecules that are there in the living cell or living organism. Okay, then we had defined a term that is the cellular pool, the sum total of different types of biomolecules, compounds, ions present in a cell. Then we had talked how can we determine the chemical composition of the cellular pool. So we had talked about the process for organic compounds and for inorganic compounds, and we have also compared the elemental composition of the earth's crust with the human body, right? So a very important diagram that is given in NCERT. Now we can move on to the page number 2. Then I had taught you about the classification of biomolecules. We have biomicromolecules, macromolecules, the examples. Then I had told you that the lipids, they are not strictly biomacromolecules. I had given a reason behind this as well. So a very important point. Now the average composition of the cells. So water 70 to 90%, protein 10 to 15%, carbs 3%, lipids 2%, nucleic acid 5 to 7%, and ions remaining 1%. Okay, so this much we had covered in the last class. Then I had started teaching you about the metabolites, so intermediates and the end products of biological metabolism. Then what are primary metabolites, what are secondary metabolites, and some very important examples given in NCERT. Then we had started with the very first biomacromolecule, that is carbohydrate. So I had taught you that carbohydrates are polyhydroxyaldoses or ketoses and the condensation products. We had talked about the classification for carbohydrates. So we have small carbohydrates and complex carbohydrates. For the monosaccharides, oligosaccharides, polysaccharides, we had defined the term monosaccharides, the general formula CnH2nOn, right? Then we were talking about the structures for monosaccharides. Okay, so structure may we have like the pentose and hexose sugars that we had talked about. Okay, so you can have the ring structure or you can have the open chain structure. We had talked about the structure for glyceraldehyde, dihydroxyacetone, ribose, deoxyribose. Today we'll be starting with the structure of glucose, right? So for glucose is a hexose sugar, it is an aldose sugar, so it will have aldehyde at its functional group. So now we are aware that for glucose N is going to be equal to 6. N is equal to 6. The number of carbon atoms is going to be equal to 6. So let's draw 6 carbons first. 1, 2, 3, 4, 5, 6. The first is going to be your functional group, the aldehyde group, right? Now for the second carbon, you are going to have OH on the right side. For the third carbon, you are going to have the OH on the left hand side, then OH. We are going to draw the D form. So OH, and here you have the CH2OH. I'll satisfy the valency of carbon here. Yes, so this is the open chain structure for glucose. Now what you have to remember that at carbon number 3, the orientation of the OH is going to be on the left hand side. Okay, so this is very important. Now I'll tell you the last chiral carbon, the hydroxyl group attached to the last chiral carbon, right? This will determine that it is D form, right? So this is the D form of glucose, D-glucose, and this is the point that you have to remember, that on the carbon number 3, the OH is going to be on the left hand side. Now I'll tell you the reason behind this. So simply, if you want to understand why at the carbon 3 the OH is on the left hand side, you will have to understand the concept of isomerism. So I would like to tell you about what is stereoisomers. So stereoisomers, the isomers having the same having the same molecular formula, having the same arrangement of atoms, but they differ in their spatial arrangement of the atoms, right? How the atoms are organized in their 3D structure. So the sequence of the atom is going to be the same, the arrangement is going to be the same, the molecular formula is going to be the same, but they will differ in the orientation of the atoms constituting that particular molecule. So this stereoisomers, right? So when n is equal to 6, I'm talking with respect to the hexose sugars, right? So you will have 4 chiral carbons. If you can even see, look at here, I'll put a star on the chiral carbon. So this is the chiral carbon, this is a chiral carbon, this is a chiral carbon, this is a chiral carbon, right? So here you have 4 chiral carbons. So the number of stereoisomers is going to be the number of stereoisomers is given by 2 raised to the power n, where n is the number of chiral carbons. Okay, so here it will be 2 raised to the power 4, that comes out to be 16. So you will have 16 stereoisomers. So out of these 16 stereoisomers, you will have 8 D isomers and you will have 8 L isomers, right? So the structure that I have drawn in front of you is of the D glucose. Now I'll just explain you this thing. Let's say if I'm drawing this particular structure, C double bond OH, 1 2 3 4 5 6. I'm making the D form. So here, right? So the sugar that I have drawn in front of you, it is D-allose. This is D-allose, right? This is D-allose. Similarly, let's say I will rotate the I'll switch the position of the hydroxyl group at carbon number 2. I'll give it, that is going to be in stereoisomer, right? So it will have some other name. If I am moving the hydrogen of the hydroxyl group on the left hand side, it will be called as glucose. Okay, so if I'm switching both the hydroxyl group at carbon at carbon 3 and at carbon 4, it will be called as galactose, right? So it will be called as galactose. Let's I'll tell you. For example, if I'm rotating this bond, so if I put it like in this manner, now it will be called as galactose. So here we have drawn the structure for galactose. So this will become D-galactose. So it's all about the arrangement of the atoms, like I should say not the arrangement but the orientation of the atoms in the 3D space. Okay, so you simply need to remember that these are stereoisomers. So if you want to understand this thing in a very good manner, so you will have to read about stereochemistry. That's a part that will be covered in chemistry, right? So I'll suggest you to read it on Google or maybe from some chemistry books. Then you will have a better understanding of the concept. Right now you just need to understand, you just need to remember the structures. If you are unable to get the concept of stereoisomerism, I can completely understand. So just remember that when I'll have to draw the structure of glucose, I'll have to put the OH on the left hand side, right? And when I'll have to draw the structure of galactose, I'll have to put OH on the left side for both carbon number 3 and carbon number 4. Carbon number 3 and carbon number 4, right? So this is very important. Now we are done with this part as well. So glucose, galactose, 2 sugars I have covered. I'll put the name here as well. So this is the 5th structure. Let me just shift this. This is galactose. Now we have drawn the open chain structure for both glucose and galactose. Next is going to be the next step is going that we are going to draw the ring form structure for both glucose and galactose. So this is again very easy. This is very easy. What you have to do is, I'll just tell you, I'll just copy this structure first. C double bond O H 1 2 3 4 5 6 CH2OH here, OH H OH OH. Okay, so this is D-glucose, the structure that I have drawn. What will I do? I will have to establish a bond between the carbonyl carbon and the carbon number 5. Okay, so what can I do is, I will remove the hydrogen, I'll shift it to the carbonyl carbon. So let's erase this here. I'll put it like this, OH. I'll remove this thing from here and I'll establish a bond. O, this, like this, right? So now I can draw the ring structure. So it is going to be a pyranose structure, right? So hexagon ring, correct? So again, you can have the alpha form and you will have the beta form. So the alpha form and beta form, they are called as anomers. If you want to get into chemistry, alpha form and beta forms together, they are called as anomers. ANOMERS, anomers. Okay, so if I have to draw, let's say, the alpha structure, I'll have to put OH on the lower side. So I'm making the alpha D glucose, right? I'm making alpha D glucose. So here, now what I had taught you in the previous session, the groups on the right hand side, right? The groups on the right hand side, you will have to put them on the lower side of the bond, right? So see this OH, I have put it here. Then I have hydrogen, I have OH. Okay, then I'll have to put the groups on the left hand side. So H, OH, H. CH2OH being a bulkier group, you will have to write it above and then you will have to write the hydrogen here. Okay, so I'll remove these tick marks and done. Here is the ring form structure for alpha D glucose. So easy, right? Similarly, I can draw it for the molecule for galactose. Galactose ke liye, for galactose, what you will have to draw again, will make the pyranose ring. Will make the pyranose ring. Let's say this time I'm making beta galactose, beta D galactose. Okay, so since I'm making beta D galactose, I'll put OH on the upper side. Okay, so B up, OH on the upper side, hydrogen here. So here I'm drawing beta D galactose. Again, the groups on the right hand side, these one, the groups on the right hand side, I will have to put them on the lower side, right? And for the left hand side one, I'll write them on the upper side of the bond. So here I will have the hydrogen, here I will have the hydroxyl groups, and CH2OH being the bulkier group on the upper side, and hydrogen here. So here is the ring structure, rings form structure for beta D galactose. Again, so simple, right? So this is how you can remember the structures, right? You will have to look for a pattern, you will have to memorize some words, and then you will be able to draw it. So done for the glucose and galactose. We are done till here. Let's move on to the next. So we have done aldohexoses, glucose, galactose, two important one, right? So as I told you that there are going to be 16 stereoisomers, right? So out of 16, 8 are D, 8 are L. So D glucose, D galactose, and I also I had also shown you the structure for allose, D allose. So 6 structures out of 16 we have covered. I had drawn the D form. So you simply will have to rotate the you will simply have to switch the position of the last you will have to switch the position of the hydroxyl group attached to the last second carbon to the left position, and you will obtain the L form, right? So very easy. Out of 16, I taught you how to draw the structure for 6 stereoisomers, right? Yes, 6. So 10 are left. It is not a part of your syllabus. Well, these structures are taught at the level of graduation or maybe above that. Okay, so we just have to stick to NCERT. So you simply need to learn the structure for glucose, galactose, and fructose, right? So the next one is going to be the keto hexose sugar, that is fructose. Okay, so 5. Let's continue with the sixth one. Sixth one, that is fructose. Correct? Fructose. So again, fructose is a keto hexose sugar. So the N is going to be equal to 6, but since it's a keto sugar, so I'll have to put a ketone group. Okay, so CH2OH, C double bond O, so here you have the carbon chain for fructose. Again, for fructose you have the hydroxyl on the left hand side. Then since I'm drawing D fructose, so I'll put the hydroxyl on the last second carbon towards right, and then I'll have to just satisfy the valency of carbon. So this is the open chain structure for fructose. So fructose, it exists in the fumarose structure, right? So fumarose structure, so that's why I'll have to draw the fumarose structure for fructose. If I want to show you how the bond gets established, it's very easy. Okay, so you will have to remove the hydroxyl from here. So OH right over here. See, this much easy. Correct? So again, I'll make the pentagon fumarose structure. So a pentagon. Let's make the bonds. Okay, so you will have to look at the position of the hydroxyl group if you want to make the alpha fructose or beta fructose. Let's say I'm drawing the alpha form of the fructose, so I'm making alpha D fructose, alpha D fructose, alpha D fructose. So OH is going to be on the lower side, so CH2OH here, then right one HOH. Here it will be OH, and here again you are going to CH2OH. This is the structure for fructose. This is the ring structure for fructose. Very easy. So I'll show you the numbering as well. So here you have the carbon number 1. This is the carbon number 2. Okay, this is the carbon number 2. This is the carbon number 3. This is the carbon number 4. This is the carbon number 5. This is the carbon number 6. Correct? Easy. So alpha D fructose. So we are done with the monosaccharides. We have learned how to draw the structure for triose sugar. Then I taught you about the pentose sugar and then the hexose sugar, the most important one. Correct? So I'll just show you the structure for glyceraldehyde, dihydroxyacetone, ribose, deoxyribose, then for glucose here, then for the galactose, this one, and the fructose, this one. Right? Easy to remember. Let's move on to the next slides. So now oligosaccharides. What are oligosaccharides? So these are again small carbohydrates. Remember, these are small carbohydrates which are formed by the condensation of 2 to 9 monomeric units or 2 to 9 monosaccharides. Right? So what are oligosaccharides? If I have to write the definition, oligosaccharides can be defined as small carbohydrates, formed by condensation of condensation of 2 to 9 monosaccharides. 2 9 monosaccharides. Okay, so 2 to 9. So disaccharides, if there are 2 monomers, trisaccharides, tetrasaccharides, pentasaccharides, hexasaccharides, hepta, and so on. The numbering will go on, right? So the most important category that we have to deal with is the disaccharides. Okay, when you have 2 monomers. So I'll put the next category, that is of the disaccharides. So it comes when you have 2 monomers, the sugar, the resulting sugar will be called as a disaccharide. Disaccharides. Okay, so disaccharide, that means you will have the 2 monomers. So there are 3 most important disaccharides that you have to remember for your syllabus. Okay, so the first one is maltose. The second one is lactose, and the third one is sucrose. Okay, so I also write about their respective monomeric units. For maltose, you have alpha D glucose plus alpha D glucose alpha D glucose. For lactose, you have beta D glucose plus beta D galactose. For sucrose, you have alpha D glucose plus beta D fructose, right? Clear? So here we have all the 3 respective disaccharides. Now I draw the structure for maltose, and then you will be able to grasp how to draw the structure for disaccharides and what are glycosidic linkage. I'll tell you about this. Okay, so after this we'll talk about what are reducing sugars. Let's move this topic aside. For maltose. So as I said that maltose, so the 2 constituting units are alpha D glucose plus alpha D glucose. So again, let's draw the structure for alpha D glucose first. Okay, so alpha D glucose alpha form, so OH is going to be on the lower side, correct? Then we have the glucose. So for the third one, the hydroxyl group is going to be over here, CH2OH, CH2OH, here OH here, OH, H, H, H. Simple. Okay, so I have drawn 2 units of alpha D glucose. So this is alpha glucose, this is again alpha glucose, correct? So now what happens when 2 glucose units are condensed together? So the condensation, that there will be the removal of water molecule, okay, and a bond will get established. So this is the carbon number 1 for maltose, for this unit I'm assuming, and this is the 4th carbon. This is the 4th carbon of the adjacent glucose subunit. Okay, so you will have to establish a bond like this. So there will be the removal of the water molecule, okay, so, and you will get the structure for maltose. So here I'll draw the resulting diagram. Okay, so this is the resulting structure. So if you can see, for this one, the carbon number 1 has got involved here. It is 4. So it is alpha 1 4 glycosidic linkage. The bond that has been established, it is the glycosidic bond. Okay, so glycosidic linkage, or alpha 1 for glycosidic bond. Okay, and here there will be removal of water. So there will be removal of the water molecule. Okay, I hope this part is clear to all of you. I hope this is clear. Clear? I hope it is. Okay, so now for the sucrose, I will not suggest you, for lactose and sucrose, I will not suggest you to remember the structures. Rather, I'll suggest you try to draw the structures on your own. You can simply Google them out and you can check whether you have drawn the right structure or not. Then very important point, that for maltose you have to remember the structure. If you are unable to remember the structure for galactose and sucrose, so you have got the logic how you can draw them. Else you don't have to worry about it. Just remember the structure for maltose. Now I'll tell you what are reducing sugars. So what are reducing sugars? Reducing sugars, the sugars that can actually reduce cupric ion to cuprous ion, right? So they can reduce cupric ion to cuprous ion. They are considered as reducing sugar. So for checking whether the given sugar is reducing or not, like two very popular solutions are the Fehling solution and the Benedict's solution. Okay, so you have the Fehling's test and Benedict's test to know whether the given sugar is reducing or not. Okay, so I'll just write the name of the solutions. It is Fehling's solution and Benedict's solution. So these solutions basically contain free cupric ions. If the sugar will be reducing, it will reduce the cupric ion to cuprous ion and you will get a precipitate. Correct? Now very important point. For a sugar to be a reducing sugar, it should have a free aldehyde group or a free keto group, right? So all monosaccharides, they have either a free aldehyde group or free keto group. So all monosaccharides are going to be reducing sugar. Okay, so I'll write it, all monosaccharides are reducing sugars. See, now let's talk about disaccharides. So for disaccharides, you can look at the structure for maltose. So here you have the structure for maltose. I'll just do the numbering for you. So this was 1, 2, 3, 4, 5, 6. Here 1, 2, 3, 4, 5, 6. Right? So if you can see that where were the aldehyde groups? So the carbon number 1. So here you have an aldehyde group, but it is bonded. But here if you will see, for this particular subunit, the aldehyde group is free. Right? So maltose is a reducing sugar. Galactose is also a reducing sugar, but lactose is a reducing sugar, but sucrose is a non-reducing sugar. Sucrose is non-reducing. Okay, so I'll write here maltose let's write and lactose reducing, sucrose non-reducing. Now, how a question can be framed with respect to NSEJS? Now, I'll tell you. So, a question can be framed in a manner that a solution was taken having free cupric ion or C2 positive ion, plus 2 ions, right? So now three sugar solutions were added to three samples of the XYZ solution. So for two solution a precipitate was observed, but for the third for the third sample, for the third sugar, there was no precipitation observed. So what can be the possible, which one can be the possible sample number 3, right? The third sugar, you have to identify the third sugar. So obviously if the options are like maltose, lactose, sucrose, and then you have galactose. So I'm just saying to complete the fourth one. So the fourth one, galactose, is a monosaccharide, so it is going to be reducing. Lactose, glucose, they are reducing, but sucrose is non-reducing. That's why it wasn't able to reduce the cupric ion to cuprous ions. That's why no precipitate was observed. So this is how the question can be framed. So whatever I'm teaching you, it's the theory part, and then we'll practice some questions related to this in the upcoming session, so you can get to know how to apply the concepts that we have learned throughout the journey. Correct? So here I'm done with the oligosaccharides. Oligosaccharides mean simply you have to learn about the disaccharides. Let's come to the next topic, that is the polysaccharides. So polysaccharides, again let's define the term first. So what are polysaccharides? So they are complex carbohydrates. What are they? They are complex carbohydrates. So I'll write this. They are complex carbohydrates formed by polymerization of large number of monosaccharide monomers of monosaccharide monomers. So in the previous table I had told you that n is going to be above 10, right? So n is going to be greater than 10, right? So more than 10 monomers will be there. Okay, so since they are long polymers, long changed polymers, so they are also called as glycans. Polysaccharides, they are also called as what? They are also called as glycans. So instead of writing a polysaccharide, if I write it as a glycan, it's going to be correct. So okay, so the polysaccharides are also called as glycans. They are long chain. So now just understand this thing. Okay, so before that, let's talk about the classification of polysaccharide first, and then I'll tell you whether the polysaccharides, how can you identify which part is reducing and which is not. Okay, so on the basis of the constituting monomers, polysaccharide can be homopolysaccharides or you can have heteropolysaccharides. Okay, so you can have polysaccharides. So you can have homopolysaccharides or you can have heteropolysaccharides. Heteropolysaccharides. Now I'll define the terms with the help of the visual representation, right? So for the homopolymers, let's say you are going to have the same type of monomeric unit, right? You will have the same type of monomeric unit, right, and so on. But for the heteropolymers, let's say you will have multiple types of monomeric units. Okay, so I can draw it with I can draw the monomers with different balls, like with different colors. Okay, so having a blue component, then you are having a red component, then you are having the green component, again repeated. Okay, and then so on, right? So homopolysaccharides and heteropolysaccharides, let's talk about the homopolysaccharide first. So as the name says, they will be made up of only one type of monomer. Okay, so the homopolysaccharides, they have two functions. They have the storage, the function of storage, and they have the function of structural support, right? They provide structural support. So you have storage homopolysaccharides and you have structural homopolysaccharides. Okay, so homopolysaccharides mean you have the storage, homopolysaccharides, and then you have the structural homopolysaccharides. Okay, so these are the functions for homopolysaccharides. Now one very important point, polysaccharides, they are large, they are complex, and they show very poor solubility in water. So I can say they are insoluble in water. What are they? They are insoluble in water, and they are not sweet to taste, right? Unlike sugars, polysaccharides or glycans, they are not sweet to taste. Starch, if you'll taste it, it will not be sweet to taste. When you chew roti, right, chapati is a very rich source of starch, right? It's a very good source of starch. So when you chew it, you have the enzyme salivary amylase. That enzyme basically breaks down the starch into maltose, dextrin, alpha maltose, dextrin, right? And that's why you get the sweet taste. Okay, so on their own polysaccharides don't have any taste. They are tasteless, please remember. Okay, and again, since they are large, they are insoluble, so they don't alter the osmotic potential or the water potential of the cell, and that's why they are meant for storage, right? Why a plant cell would like to store its or an animal cell would like to store like the monosaccharides in the form of starch or glycogen. So the reason is this, that these molecules, these are polysaccharides, they are very large in size, they are complex in the structure, they are not soluble, and that's why they are insoluble, they are stable, and third, they don't alter the osmotic potential of the cell. To alter the osmotic potential, the substance has to be soluble, but since they are insoluble, right, or they have very poor solubility, so that's why they don't alter the water potential, and that's why they are ideal for storage purposes, right? And since they are large, they are complex, so obviously they can also be served for structural purposes. So I'll give you the example of storage. The major examples given in NCERT, first is glycogen, then you have starch, and then you have inulin. Inulin, don't get confused with insulin. Insulin is different. It's a hormone. Beta cells of islets of Langer, and insulin, that is a protein made up of 51 amino acids. Inulin, it is a homopolysaccharide made up of fructose subunits. Okay, so here you have fructose, but there you have amino acids. Okay, so storage. For the structural one, we have two examples. First one is of cellulose, chitin. The examples given in NCERT only, and even if you'll refer to some extra books, only these examples are mentioned. Okay, and then for the heteropolysaccharides, so they only serve structural purposes. They don't have purposes of storage. Okay, so here you only have structural purposes. Okay, so they are always structural. So it's structural heteropolysaccharides. Now, structural heteropolysaccharides mean the examples. So the examples are going to be peptidoglycan, and then you have hyaluronic acid. Okay, so so according to NCERT, we just have to learn about glycogen in detail. For starch, inulin, I'll just tell you the main points. Same goes for cellulose and chitin, and peptidoglycan and hyaluronic acid. So peptidoglycan we'll talk about in depth when we'll do the structure of a prokaryotic cell, and hyaluronic acid. So hyaluronic acid, yes. So hyaluronic acid, I'll tell you some important points right now. and hyaluronic acid, it is basically a major constituent of the ECF, extracellular fluid. Okay, so इनके बारे में for peptidoglycan and hyaluronic acid, I will tell you right over here only. Okay, so peptidoglycan, since it is going to be a heteropolysaccharide, so it is made up of NAG and NAM, N-acetyl glucosamine and N-acetyl muramic acid. So you have alternating units of NAG, NAM. NAG, N-acetyl glucosamine and N-acetyl muramic acid. Hyaluronic acid के लिए you have N-acetyl glucosamine, NAG, plus glucouronic acid. You have glucouronic acid. Glucouronic acid. Okay, so glucouronic acid, it is gluco. Okay, so CU directly. Glucouronic acid. Okay, the pronunciation is so bad. So peptidoglycan, it is found in the bacterial cell wall. All right. Major component of bacterial cell wall. Hyaluronic acid, extracellular fluid, ECF. Vitreous humor of eye. Vitreous humor. It is a component of synovial fluid. Synovial fluid. You want more examples? CSF, cerebrospinal fluid, CSF, etc. These are the places where you can find the hyaluronic acid. In cartilage of bones and the cartilage of matrix of cartilage. I'm really sorry. Matrix of cartilage matrix. Again, you will have hyaluronic acid. So yes. Now come over here. So let's start with glycogen. I'll tell you a little depth about these, but these one are done. Okay, the hetero one are done. Let's start with glycogen first. We are starting with glycogen. Okay, so glycogen may first of all where it is found. So it is again, it is a storage homopolysaccharide. It is a storage homopolysaccharide for animals, bacteria, fungi. So when I say animals, including human as well. Glycogen is also known as animal starch. It is also known as animal starch. Next, so animal storage homopolysaccharide for animals, bacteria, fungi, it is also known as animal starch. Now we'll talk about structure. Acha, where it is stored in human body. So glycogen is stored in human body at two places, liver and muscles. Okay, so clear? Okay, I'll also write the points: animal, bacteria, fungi, storage homosaccharide for animals, bacteria, fungi, also known as animal starch, and stored in humans. Correct? Liver and muscles. Now let's talk about the structure for glycogen. Very important about the structure for glycogen. So glycogen, first of all I'll tell you, glycogen, so it is a polysaccharide, right? It is a homopolysaccharide. So the monomeric units are alpha D glucose. So monomeric unit, that is alpha D glucose. So how many monomeric units will be there for one molecule of glycogen? So approx 30,000. Okay, so 30,000 glucose residues you will have in one molecule of glycogen. Okay, so what will be the molecular weight? So the molecular weight is going to be somewhere around 4.8 million Dalton. Now in some books you can find it as like 3.5 million Daltons. So the data varies from book to book. Okay. 4.8 million Dalton. Okay, so let's talk about the structure. Structure is very important. So here, what you have, first of all, you have a straight chain. You have a straight chain arranged in a helical manner. Okay, so you have a straight chain arranged in a helical manner. Okay, so you, first of all, what you have in this structure, we are talking about the structure of glycogen. So you have the straight part, you have a straight chain that is helically twisted, right? Helically twisted, right? So since it is twisted, so it is going to have turns, so each turn will have 6 glucose residues, right? So each turn, one turn will have 6 units of glucose. Very important. Now next point, that you will have the side chains. Second component is going to be the side chains. So second, the side chains. So occur at every 10 to 14 residues, occur after every 10 to 14 residues of the straight chain. Okay, so 10 branching at 10 to 14 residues. Okay, so after every 10 to 14 residues you will have a branching. So how the ultimate structure will look like, it will be like a spiral. Okay, and then you will have the side chains like this. Okay, so this is how the structure would actually look like. So now the next point, that does the glycogen show color with iodine? So the answer is actually yes. The helical, like the helical structure that you see over the screen, this helix, so the iodine molecules will get trapped in this helical structure. So yes, glycogen also show color with iodine. It shows like reddish color. Glycogen shows reddish color with iodine. So now what is given in NCERT, that is actually like the structure that has been described in NCERT, it is actually little wrong. Second point, I don't think they have mentioned about the color with iodine. So I'll request you to either refer to some very good books or some research papers. You will find this data. Okay, so reddish color with iodine. Okay, iodine, correct. So this much information you need to know about glycogen. Done. Let's move on to the next one, that is starch. I can write it here. Second, that is starch. So starch found in plant storage homopolysaccharide for plants. Again, the monomeric unit, it's going to be alpha D glucose. Okay, so I'll write monomeric unit. That is again alpha D glucose, right? Correct. Clear till here? So now we'll talk about the structure for starch. So starch actually consists of 2 components. You have amylose and amylopectin. So we are talking about the structure. So starch, it comprises of amylose and amylopectin. Amylose and amylopectin. For amylose, what you will have, so for amylose, like each molecule of amylose will have around 200 to 1000 glucose units. So I'll write 200 to 1000 units. For amylopectin, so one molecule of amylopectin will actually have 2000 to 2 lakh, yeah, 200,000 or simply 2 lakh units, right? Okay, so for amylose, what you have, so for amylose you have again the helically arranged straight chain. There is no branching for amylose. Okay, so no branching for amylose. It is helically, and it is right-handed, like it is right-handed twisted. Okay, so it is right-handed helical. So it is right-handed helix. Better, right? It is a right-handed helix. Amylopectin, so straight chain with side branches. So for amylopectin, again you have tiny, like you have the helices. So amylopectin, the amylose, it shows dark violet color with iodine. Amylopectin also gives red violet color with iodine. Okay, but the color for the amylose is so intense that the amylopectin color derived from amylopectin doesn't show up that much. Okay, so again NCERT may something different is given. I'll request you to please refer to some either very good books or some research papers. You will find this data. So right-handed helix, here it is straight chain. The straight chain will again have some helices with branching, with side chains. This branching will occur about 20 to 24 or 25 residues. So I'll write this 25 residues. So the branching will happen at about 25 residues. Correct? So this much information you need to have. Okay, so okay, I'll also write the color with iodine, right? So dark violet color with iodine, dark violet, and here it is red violet with iodine. Right? So one very important point, applicable to both glycogen and amylopectin, that, like, see, if I have to draw the structure like this, so this is the straight chain. Here you have the glucose subunit, right? And this here you have the branching. So let's focus over here about these three subunits. Okay, so यहाँ पे you have glucose, So this linkage between two adjacent subunits, it is going to be alpha 1,4 glycosidic linkage. Alpha 1,4 glycosidic linkage. And here you have alpha 1,6, the branching. It is alpha. Okay, so again here you will have glucose. Okay, so here it is alpha 1,6 glycosidic linkage. This much data you need to remember. Okay, so this part is very important to know. You must know these details. Next, so we are done with glycogen, we are done with starch. Let's talk about inulin. So inulin, again, it is storage homos polysaccharides in some of the plants like dahlia and related plants have inulin. The monomeric unit, it is a fructan, right? So it is fructose. So for glycogen and starch, it is alpha D glucose. Glycogen and starch both are branched polysaccharides. Inulin, so inulin, it is fructan. Or the units are fructose. The monomeric unit is of fructose. Okay. Now one very important point about inulin, that inulin does not get metabolized in human body. So inulin is used for the functioning test for organs like kidney, right? So kidney function ke liye, because it must be filtered through the kidney into the urine, because it is not metabolized in the body. So that's why inulin is used for the functioning test for kidney. Next is here the cellulose, and then we have chitin. So cellulose, it is an unbranched polymer. The monomeric unit is going to be beta glucose. It is beta glucose. And for chitin, it is NAG and NAG. And acetyl glucosamine, chitin, found in the exoskeleton of the insects. Let's say fungal cell wall, you have chitin. Cellulose in the plant cell wall, cellulose. Okay, so this much data you have to remember. Okay, now one very important point, that the right side of let's say starch or glycogen for a polysaccharide, it is going to be the reducing end, and the left hand is going to be the non-reducing end. So I'll write right end is reducing and left end is non-reducing. Okay, so let's move ahead. So I hope the notes that I am sharing with you, they are worth noting down. Okay, so let's move ahead and let's start with the next topic, that is of amino acids. So what are amino acids? So amino acids, they are basically substituted methanes. NCERT writes that amino acids are substituted methane. Okay, so I hope everyone is aware about the structure of methane. It is CH4, the formula is CH4. So here you will have hydrogen, R group, R can be any side chain, COOH, and here you have NH2. Okay, so if you can see, so this is the alpha carbon, like this is the functional group, COOH, like the primary functional group, then this is the alpha carbon. Okay, so these are also alpha amino carboxylic acids. So you can also write that this is the general formula for amino acids. So they are also alpha amino carboxylic acids. It's what are amino acids? So they are alpha amino carboxylic acids. Now the amino acids, they are the monomeric unit for proteins. Okay, so they are the monomeric units for proteins. Monomeric units for proteins. So I'll tell you a very important point about amino acids, that there are 20 fundamental amino acids. The fundamental amino acids, that is coded by the generic code, right? Coded by the DNA. So 20 fundamental amino acids are there, but these fundamental amino acids, they further go post-translational modifications and they get converted into many derived amino acids. So the number of amino acids as such is 100 plus, like hundreds of derived amino acids are there, but they are derived from the basic 20 fundamental amino acids. So this is there is a difference. There are 20 fundamental amino acids, and after post-translational modification, hundreds of derived amino acids get synthesized. Okay, so very important point, that there are 20 fundamental amino acids. When I say fundamental, that is coded by the genetic code, right? Coded by the DNA. So 20 fundamental amino acids are there. Okay, now the proteins made up of multiple amino acids. Now a protein, like the proteins, they tend to have multiple amino acids. Okay, because the role of the proteins the role of the proteins is so crucial that, and because it has to undergo unfolding and everything, that's why the proteins, they are always heteropolymers. They are always heteropolymers. Heteropolymers. Proteins are always heteropolymers. Okay, so proteins are always heteropolymers. Now, so we have learned about what are amino acids. They are the monomeric units for proteins. There are 20 fundamental amino acids. They are substituted methanes, or the amino acids are substituted methanes or alpha amino carboxylic acids. Now, the next important point, that is the peptide bond. So, what is peptide bond? So, the bond established between, or the bond linking two amino acids will be called as the peptide bond. Chemically, it is a amide bond only. Okay, so if I have to draw, let's say okay, so if I have to draw a bond over here, yes, so this is how a bond can be established. This will result in something like this. So here you have this is the peptide bond. This is the peptide bond. Okay, so this is the peptide bond. There will be obviously removal of water. So a molecule of water will get removed. Correct? So now you can see, if you have a long chain of amino acids, you will have the N terminal, you will have the N terminal, and you will have a C terminal. So at one end, NH2 group will be unoccupied, and at the other end, at the opposite end of carboxylic, there will be a free carboxylic acid group. This is the C terminal. Okay, so very important when it comes about the polarity of the polypeptide chain. Let's move ahead and let's talk about the classification of amino acids. Classification of amino acids. Types of amino acids. So I'll make a flow chart for you, and then I'll also tell you about the structures of the amino acids, and I'll request you, if you can remember the structures, they are very easy to memorize. So if you can remember the structures, it's well and good, but remember it. For at least I'll tell you for the amino acids for which you have to remember the structure, right? So types of amino acids. So you can have, rather I'll make it in this manner. Visible, yes. You can have neutral amino acids. Some will be acidic amino acids. There will be some basic amino acids. There will be some sulfur containing amino acids. Okay. Then I'll pull another branch. There will be some aromatic amino acids. Okay, so here you have aromatic amino acid. You will have alcoholic amino acid, and then you will have heterocyclic amino acid. Heterocyclic amino acids. Okay, so this is the classification. Right now I'll distribute those 20 amino acids into these categories, right? So let's start with the neutral one. So it is glycine, alanine, valine, leucine, isoleucine. Okay, so these are glycine, alanine, valine, leucine, isoleucine. These are neutral. These are neutral amino acids. Then you have acidic one. So acidic में aspartic acid, asparagine, glutamic acid, and then glutamine. These four. Okay, so aspartic acid. So this is the acid, and then you have its amide form. So aspartic acid का the amide form is asparagine. Asparagine. Then you have glutamic acid, and then you have glutamine. Okay, so aspartic acid, asparagine, glutamic acid, glutamine. Then basic में, so arginine and lysine. So asparagine is different, arginine is different, right? Arginine, lysine. Okay, so now sulfur containing two very popular, cysteine and methionine. Cysteine, methionine. So this is cysteine, not cytosine. Please remember. Okay, so cysteine and methionine. Then let's talk about the aromatic one. So you have for the aromatic one, you have phenylalanine, you have tryptophan, and you have tyrosine. You have tryptophan for the aromatic. Now alcoholic में, so you have serine and threonine. Here you have serine, threonine, and for the heterocyclic one you have proline. And one more, that is histidine. So let's count 1, 2, 3, 4, 5. 5 neutral, glycine, alanine, valine, leucine, isoleucine. 1, 2, 3, 4. 5 plus 9, 5 plus 4, 9. Acidic में aspartic acid, asparagine, glutamic acid, glutamine. Then 2 for basic, so 11, 9 plus 2, 11. Arginine, lysine, sulfur containing plus 2, so 11 plus 2, 13. Cysteine and methionine. Then for aromatic, you have 3, that is phenylalanine, tyrosine, tryptophan. Alcoholic, serine, threonine. 2, 5 plus 3 plus 2, 5. Heterocyclic, proline and histidine. 7 plus 13, that comes to 20. So the here are the names of the 20 fundamental amino acids. Now let's look at their structure. So basically they all differ in their side chain. So I'll request you, if you can remember the diagrams, else I'll tell you the most important one. So glycine, alanine, valine. So glycine is important. Alanine में you only have the CH3 group, and then I'll suggest you to remember the structure for proline. Okay, so this one is proline. This one is proline. So if you can simply Google out, you will be able to find the structures for all the amino acids. Okay, easy peasy, right? So that this is all about the amino acids. Now let's talk about the proteins. So the amino acids, they get condensed. There is the peptide bond formation and the proteins are formed. Okay, so now we need to know about the level of organization of proteins. So first of all, the proteins, they have the primary structure. What is the primary structure for a protein? So the sequence of the amino acid, the linear chain of amino acid will be called as the primary structure of the protein. So if you can see the amino acids, here you have the only the chain of the amino acids. So this is the primary structure for a protein. Then this primary structure, so there, here, as you can see, here you have some acidic residues, some basic residues, some neutral residues, some aromatic residues, some alcoholic residues. So these residues are able to make different types of bonds. So they will try to make some bonds among themselves, the amino acids, and then the secondary structure will originate. So secondary structure may the hydrogen bonding, the primary bonding is the hydrogen bonding. So the hydrogen bonds get formed either within the same chain or between the chains, that gives rise to the secondary structure. So you have the alpha helix and the beta pleated. So let's know about the secondary structure later. So aware of the primary structure, linear sequence or the linear chain of the amino acid. Then comes the secondary structure. So for the secondary structure, you have either the alpha helix or beta pleated. Alpha helix and beta pleated. So for the alpha helix, you have the hydrogen bonding within the same chain at ith and I plus 4th residue. So hydrogen bonding between ith and I plus 4th residue. Okay, so with every 4th residue you will have the bonding, the hydrogen bonding. So this will give rise to a helix this way. Okay, so a helix will be found. Better, I have already inserted the diagram. So you can see the helix will get formed. Okay, so the hydrogen bonding is happening within the same chain. For beta pleated, you can have two: parallel beta pleated structure, or you can have the anti-parallel beta pleated structure. So you can have the parallel or anti-parallel beta pleated structure. Parallel or anti-parallel. Now what does it signify? So parallel mean, like, let's say you have two chains. So this is going to be, so the bonds that I have drawn, these are the hydrogen bonds. Okay, so N terminal, N terminal, C terminal, C terminal. So this is the parallel beta pleated structure. Two chains is held together by the hydrogen bond. For anti-parallel, it will look something like this. Okay, so N terminal, C terminal, C terminal, N terminal, right? So the two chains are anti-parallel. So that's it. So alpha helix, it is found in examples में ले लेते हैं. So keratin में primarily you have the alpha helix. Beta pleated में you can take the example of fibroin, which is a protein found in silk. Okay, so beta pleated structure. Then these structures further fold. Okay, and then you get the tertiary structure. So what was the primary bonding responsible for the secondary structure? Hydrogen bonding. Then comes for the tertiary bonding. So multiple bonds will be there. Okay, multiple types of bonding will be there. What will be the type of bonding that will be there among the subunits, among the amino acids? So first you can have the covalent bonds. You can have disulfide bonds. You can have electrostatic force of attraction. that is the ionic bonds. You can have van der Waals bonds, van der Waals forces, and then you can have hydrophobic interaction. Hydrophobic interaction. So these are the types of bonds that are responsible for the stabilization of the tertiary structure. So you have the secondary structure, it further folds down into the tertiary structure. Okay, so this will how it will look like. So you use in some part you can have some helix, in some part you can have the pleated structure, and that's how. Now if the protein is made up of multiple subunits, let's say hemoglobin. So hemoglobin, it has 2 alpha chains and 2 beta chains. So when a protein has more than one subunit, it will make the quaternary structure. Right? Example, you have hemoglobin, 2 alpha chains and 2 beta chains, so that will be the quaternary structure for the protein. Okay, so quaternary ka very popular example is hemoglobin. So that's all for today's class. Now with the next class, I'll start with the classification of proteins, and then we'll also cover the DNA and RNA. Okay, so in the next class, we'll talk about the classification of proteins plus the nucleic acids. Please be regular with this series, because this is going to help you build your foundation very strong for the upcoming NSEJS 2025 exam. Thank you very much, guys. That's all from my side. Good night.